I accepted the request of Dr. Pablo Engel, the president of the Scientific Committee of the Society, to give a talk in this first session of this academic year. Mostly because I feel obliged, because he has put a lot of effort to ensure that we have a good program during the course, and also he has ensured an extraordinary annual meeting by enrolling into our meeting the top researchers of the European Federation of Immunological Societies, of which work he has served for many years, and in gratitude they have accepted to come. He has completed the program, and he asked me to give the kickoff session, and I accepted also because this is my last year as a president, and is going to be also my last academic year as an active professor at the Autònoma University of Barcelona and as a consultant at Hospital Vall d'Hebron. And I thought it would be appropriate to give a talk that it would recapitulate a bit of the most interesting findings that I did as a researcher over many years. Obviously, I will not review everything because I will bore you terribly, but I have chosen a few that I think will be interesting, and they will be around the subject of immune tolerance. And at the end, I cannot avoid to mention a few things about COVID, because that has affected our community, not only in the way to work, but also by posing very interesting and difficult immunological questions. You will have please to forgive me to be in this lecture occasionally a little bit nostalgic, because obviously it covers periods, a long period, and some of the people whom I started to work have by now are not any longer alive. Why autoimmunity is important? Well, autoimmunity is important because it's an increased health problem. It now affects nearly 10% of the population. Is not that often lethal as can be cancer, but is a cause of a lot of suffering. Is composed of many different diseases. We counted about 70 different autoimmune diseases, clinically very diverse. Most of them are chronic. For some, we have some treatments. Some of them, fortunately, they remain on low. We can control the activity and remain on low activity for a long time. But most of them we cannot cure. Obviously, autoimmunity is the failure of the tolerance mechanism, and immunological tolerance is one of the most complex aspects of the function of the immune system, because it does not depend on a single element. It depends on the good harmony, on the functioning of many of the elements. In a way, we could compare to superior intellectual functions of the brain that are not localized because they depend on the harmonious function of many parts of the brain. We can locate where speech is or where the visual cortex is, but we cannot identify the area where it resides talent or it resides the superior capabilities. And I will discuss tolerance in the context of thyroid autoimmune disease, mostly in humans, because, as you see in this table, is the most prevalent, and because of that, I will have to give you, for the students mainly, who may not be so familiar with thyroid autoimmune disease, a very short introduction on what are this type of diseases. We have two main kinds: autoimmune thyroiditis, that when there is lymphomatous goiter, we call it Hashimoto. It is very common. It may affect about 4 to 5% of the population, leading to hypothyroidism, subclinical or clinical, and is due to the slow progressive destruction of the thyroid tissue. In spite of how common it is, and it was discovered quite a few years ago, there is still some controversy on which are the main pathogenic factors that lead to the destruction of the thyroid tissue, whether they are mostly CD8 cytotoxic T cells, We know that there are many T cells that contribute to the production of cytokines that can be also toxic to the thyroid follicular cells. And there is also the possibility that the antibodies to the thyroid peroxidase and the thyroglobulin and blocking antibodies to the TSH receptor or the thyrotropin receptor may also play a role. But it's not quite yet clear. And probably not in all the cases each factor plays the same role. 4/5 of people with thyroid autoimmune disease have hypothyroidism, and then about 1/5, 20%, they have another entity, which is still quite common, which is autoimmune thyrotoxicosis of Graves' disease. In this disease, we see a progressive hyperplasia of the thyroid tissue, and this is clearly due to autoantibodies to the TSH receptor. The TSH receptor is the trophic and function regulatory receptor that is on the thyroid follicular cells. So these people have autoantibodies that mimic the effect of the physiological hormone, the TSH that is normally produced by the pituitary. And as a result of the stimulating antibodies, they develop hyperthyroidism. So they produce high levels of T4, TSH gets reduced by negative feedback, but because TSH receptor, it is expressed at a high level in the thyroid, but it is expressed at low levels in other tissues like adipose tissue and in the tissue periorbital of the eye, we have some extrathyroidal manifestation in Graves' disease like esophthalmos and predictival myxedema. Esophthalmos can be a serious problem. So why to study thyroid autoimmune disease as the model to try to understand better human autoimmune disease? Well, there are some rational and some serendipitous reasons for me. One is that it's high prevalent, so it's an important health problem, even if we have some good treatment. The autoimmune phenomena, the autoantibodies to thyroid antigens are easy to detect. We have very good methods. They are high titer. They can be well studied easily. Is one of the few diseases in which we have access to the autoimmune tissue because it is not rare that thyroid gland is removed, mostly in cases of Graves, so we can study the tissue. Logistically, it's not always easy, but we can study it. And because it is a representative effect, is a representative example of other autoimmune diseases because it is associated with the other typical, especially organ-specific autoimmune diseases like type 1 diabetes, autoimmune gastritis, autoimmune destruction of the suprarenal cortex like Addison, and even is associated to rheumatoid arthritis. So we can infer that has a similar pathogenesis. And then there is a reason, a serendipitous reason. When I joined University College in London to do my postdoc, my project was in diabetes, but diabetes, I had to do experiments with human islets, and they were very hard to get. So I had a lot of time, free time, in which I was not able to do experiments on diabetes. And I was lucky to meet Emilio Curi, who was an Argentinian fellow that was working in thyroid immunity. And he was working closely with Professor Deborah Donig and Professor Ivan Reut, who were the co-discoverers of thyroid autoimmunity. And on top of that, thyroid tissue was easy to get at the University College London because many people were operated from thyroid autoimmune disease, and it was relatively easy to culture. So I dedicated quite some time as a part-time project to the thyroid. The questions that are important to answer in autoimmunity, and which are common for most of them autoimmune disease, especially the organ-specific ones, are the identification of the predisposing genes, the identification of the trigger elements, but the core questions are the elucidation of the roles of central and peripheral tolerance in the pathogenesis. What fails in those regulatory systems that leads to autoimmunity? And once we will know that, we will be in the position to control them and even to cure them. This is the timeline of thyroid autoimmunity. Thyroid autoimmunity was first discovered by, as I said, Deborah Donig, Ivan Reut, and then also by Noel Ross in Mitesky in the US. Our English colleagues did discover the human disease, and the American group discovered experimental thyroiditis. And that was back in 1956. Soon later, people identify thyroglobulin as one of the targets of the autoimmunity. And then they discover that the other target was microsomal antigen, which was actually then identified as the thyroid peroxidase. Then they identify an activity that was capable of stimulating the thyroid gland. They call it LATS, and then we know that there were antibodies to the TSH receptor. And so it was clearly established that both hypothyroidism, Hashimoto thyroiditis, and Graves' disease were caused by an autoimmune mechanism. In the late 70s, it was recognized that a number of autoimmune diseases were linked to HLA alleles, and it was also in the late 70s that it was recognized the association of thyroid autoimmune disease with some HLA alleles. And so HLA became important. It was intriguing why these alleles were associated. And also in the late 70s and the beginning of 80s, we started to have monoclonal antibodies, and these monoclonal antibodies allowed to identify clearly which cells will express HLA class 1 and class 2 in the tissues. And it was also then a lot of discussion on how HLA will regulate the interaction between antigen and the T cells. It was by then known that T cells will require to recognize HLA on the target cells. Not exactly how that did occur was not yet known, but it was known that was a requirement. So we got interested to know whether HLA class 1 and class 2 were expressed in the target organ of our favorite autoimmune disease, which was thyroid autoimmune disease. So we did with Toshi Hanafusa, my fellow then, we were together fellows at the University College London. We got hold of some monoclonal antibodies to HLA class 1 and class 2 that were produced actually locally. And we could see that, as expected, in normal thyroid, the HLA class 1 was expressed only in the capillary. You will see them in the interstitium between the two layers of epithelium. And HLA class 2 was even expressed in less cells, only in occasional cells that we know now that they are mostly dendritic cells. We thought they were mostly macrophages. And by then, there were some people that demonstrated that HLA class 2 could be induced, at least in T cells, by activating them. So Toshi and myself thought that would be intriguing, and we asked our boss, Franco Bottazzo, and he said, yeah, this is intriguing, why not give it a go? So we gave it a go, and we cultured thyroid follicular cells. And here you see the thyroid follicular cells. When they are in monolayer, they are spread, and you can recognize, identify that they are thyroid follicular cells because they are stained with antibodies to TPO. Then we call them microsomal antigen. And interestingly, when we added an stimulus that at the time was PHA, but we know now that it's mostly interferon gamma, the responsible for this action, these cells could express HLA class 2. and we could further identify them by double exposure and so on. So then we were intrigued whether these things that we have managed to induce in vitro could be actually seen in vivo, and we look at sections of autoimmune thyroid tissue that we had in our minus 80, sorry. And we could see that in a thyroid gland that is infiltrated, you will see in the left part of the slide, that an antibody to HLA class 1 will stain intensely the infiltrating cells and less intensely the epithelial cells. It is a pity with this system I cannot use a pointer laser to point it to you, but I think it is pretty clear. When we look at the nature of the infiltrate with a common leukocyte antigen in the right lower panel, you see the intensity of the infiltration. And if we look for class 2, and even in areas that were not so infiltrated, in a case of Graves, in the upper right panel, you see that the epithelial follicular cells were intensely stained. And that happened in most of the glands. So we were really very shocked by that, because it was supposed that HLA class 2 was a molecule for presenting antigen and therefore only expressed in antigen-presenting cells. And Toshi make these drawings that are still, now they look a little bit naive, but I like them because they are very expressive, in which you see that by expressing HLA-DR class 2, the thyrocytes will become capable of interacting with helper T cells presenting antigen, and then these helper T cells could provide the help for the B cells to produce the autoantibody. So this was our hypothesis in 1983, and we published and we proposed, and it was quite welcome by many people, and others remain skeptical, as it should be in a scientific community, right? So in the last, for a number of years, apart from doing research in other fields, one of my research was into identifying what was the factor that will be capable of inducing HLA class 1 and class 2 in thyrocytes, which one of the main ones is gamma interferon. We demonstrated that these cells could express also the addition molecules that are required for interacting with T cells, like ICAM1. Also, they express NCAM. They can express in some occasions CD40 even. But then later on, it was well established that antigen-presenting cells, to present antigen to naive T cells, they need to give a second signal, and the second signal that is normally plays the main role are CD80 and CD86. And thyroid cells have not been convincingly shown that can express these molecules. So at some point we thought, well, maybe this is something that is important for chronic, for the chronicity of the disease, but not so much for the trigger. But overall, I think all these experiments contributed to establish a concept that I believe is an important concept and is well accepted, is that in the periphery, apart from the different mechanisms that operate in the lymph node, in the secondary lymphoid tissue, for generating tolerance, which is tolerogenic presentation by non-activated dendritic cells, there is also a role for what has been called immunological ignorance. And immunological ignorance is a name that describes the fact that peripheral cells, thyroid follicular cells, but for instance also muscle cells, all the parenchymatous highly differentiated cells, they are not really very much in contact with the naive T cells. The naive T cells that they contain the more broadly, the broader repertoire, and that they could potentially contain more autoreactive cells, do not patrol, do not go through the parenchyma. It is only monocytes and a few memory cells that do that. So the fact that normally parenchymal cells, epithelial cells like thyroid cells, do express a low level of HLA class 1, no class 2, and low level of addition molecules probably contributes to maintain tolerance. And I think that's a general principle that is accepted, and that now we will go into some other aspects of it. But before we go into these other aspects, we were in the laboratory working with my colleague Dolores Jara-Quemada, who joined me at the beginning of the 80s, and she had a lot of experience on working on the T cell response, and we tried to analyze the T cell response to thyroid antigens. And one of the ways to do that was to try to generate T cell clones and to generate, to measure in culture the ability of T cells to produce cytokines and to recognize antigens. And that proved to be very hard. It was not impossible, there were some results, but actually the responses were remarkably weak. And they were remarkably weak when discussing with Dolores, who had experience also in the response to transplant. It was very weak compared to T cell response, for instance, to an allograft, in which you can measure it very well. Or even to nominal antigens. So when you immunize an animal or human being with tetanus toxoid, you get a very clear T cell response. But that's not the case with autoimmunity. So we were intrigued if these autoimmune responses actually were halfway or highly regulated. And one of the possibilities is that there was central tolerance. But we were also intrigued by, when we studied all these tissues, by the highly organized nature of the lymphoid tissue in these glands. Actually, and you see in these pictures, especially in the central one and in the right hand one, is that both in Graves' disease and more prominently in Hashimoto disease, the infiltrating lymphocytes, they organize themselves in a way they look like a lymph node. And you see even the type of structure that we call germinal centers. So trying to understand a bit further what was the role of the target tissue into the development of the autoimmune process, or in the maintenance of the autoimmune process mainly, we decided to investigate whether these lymphoid follicles had the features of functional canonical lymphoid follicles that you can find in the lymph nodes. So we look at the distribution of the different types of addition molecule and of selectins, and that was very similar to the one in the canonical, and this image shows you the distribution of ICAM1 and ICAM3, which will be similar to the one in a regular lymph node. Those are in the upper panel. And we also look for the presence of the high endothelial venules that, as you know probably, are critical for recruiting naive cells into the secondary lymphoid organs, and are also what they maintain the infiltration. And indeed, we could visualize and identify with the right monoclonal antibodies that these lymphoid follicles that are inside the thyroid, they do express the molecules to attract the traffic of the naive cells. Looking for features that are typical of the functional lymphoid follicles, we look at the presence of B cells or plasmablasts that would be actively producing antibodies to the typical thyroid antigens like thyroglobulin and thyroid peroxidase. And indeed, we got help from the industry, and we got fluorescent level thyroglobulin and thyroid peroxidase label TPO. That was with the help of Pharmacia at that time, which labeled them with biotin. And we could demonstrate that there were cells that were producing IgG, and some of these cells that were producing IgG were producing IgG against thyroglobulin or IgG against TPO. It was not so easy to demonstrate, and we have not been able, I think nobody has been able to demonstrate with this method the presence of cells producing antibodies to the TSH receptor. And one relatively unexplained and still but common feature of canonical lymph nodes, lymphoid follicles, and the lymph nodes in the canonical lymphoid follicles is the expression of the RAG1 and RAG2 genes that, as you know, codify for the recombinase. And we could find that in some of these glands, in most of them, on those that had clear follicles, there was message for those RAG1 and RAG2, and that was as a feature also of functional germinal center. So the conclusion that these germinal center were active, they produce actively thyroid antibodies, and they probably contribute to the perpetuation of the disease. At some point, we were also looking for more features that can explain the autoimmunity thyroid, and the transcriptomic profiling technology became available, and we decided to do some exploratory non-hypothesis-driven experiments, and we selected some glands with different time courses in terms of the disease, and we selected glands that we know that were taken very soon after surgery, so the integrity of the RNA was maintained, which is one of the difficulties. And when we did the Affymetrix arrays, we could see that even with the unbiased hierarchical clustering, the controls and the Graves' disease tissue were quite different. And you could see as the genes that appear overexpressed, their number increases with the progression of the disease because they got spontaneously almost in chronological order by the bias clustering. And that is probably due to the progressive infiltration. The messages were not as exciting as we expected because at the end, these signatures, genetic signatures, reflected what happened at the histological level, and we could see many genes involved in antigen presentation, in chemotaxis, and in the humoral response. It was interesting to see that there was a transcriptomic signature of interferon, which it was expected, but to a large extent was not expected to be so marked, and also some genes that are normally associated with viral response, which are intriguing, and we are still intrigued by that, and it's not been worked out why are there. Another thing that was somehow new, we should have suspected that we didn't know, is that besides a signature of M1 inflammatory macrophages, there was a signature for M2 more reparative type of macrophages. And this actually, when we look at the tissue, we could see some of this M2. So this message was interesting because it tells us that one of the reasons why probably autoimmune diseases are so slow is because in spite of that we measure mostly the active autoimmune response and the high affinity antibodies, the immune system is also trying to repress, to downregulate that response. And actually what we see in autoimmune disease is the result of two competing forces, the autoimmune response and the regulatory response. And this is not only because of them two, we also, in this transcriptomic analysis, we detected quite a few genes that are together pointing to a regulatory response that tends to downplay the response. So by 10 years ago, we had a bit more of information, but still we did not have fine explanations for many of the main questions. And I can advance you that we are not much better now, but we have a few more things. For quite a few years, but mostly in experimental animals, it was well known that apart from the signal 1 and the signal 2 that trigger the activation of T cells, there are a number of signals that regulate the progression of the immune response, and that some of them were stimulatory and some of them were negative signals. And when the oncologists started to take advantage of these regulatory systems by blocking the inhibitory signals, and they realized that by blocking the inhibitory signals of the T cells in tumors in which presumably there was an immune response to the tumor, they could elicit an immune response to the tumor and induce a regression of the tumor. That, as you know, has been big news, has been the people who promoted that therapy were given the Nobel Prize, and now this type of therapy is widely used, especially in Western countries because it's quite expensive. And one of the most clear side effects of this therapy is the appearance of autoimmunity. Many of these patients develop a sort of ulcerative colitis, which normally is transient, but about 10% of them develop thyroid autoimmunity. And we were intrigued by that, and then we decided to study why was that. So we look at the situation of the PD-1, PD-L1 axis in Graves' disease, and we look at several aspects. I will not go into all the details. As expected, we found that the T cells that infiltrate the thyroid express PD-1 because they are activated cells, and PD-1 is not only, as you know, a marker of exhausted cells, it's also a feature of activated T cells. Even in the periphery of the patients with Graves' disease, there is a moderate amount of PD-1 positive cells, but it's mostly detectable in them. And if we look at the tissue, and I think one of the advantages of this presentation online is that in the screen of your computer you will see this much better than in projecting into a lecture hall. You see that we can visualize PD-1 positive cells in the infiltrating thyroid. You see the thyroid follicular cells are here identified by cytokeratin, PD-1 in red, CD4 in green, and you see the classical yellow double staining in a proportion of these cells. But what we were more interested was, well, how is PD-L1, which is the ligand for PD-1 in the thyroid. We knew from previous research that in general, PD-L1 can be induced in the tissues by interferon gamma. We knew that thyroid tissue express class 2, and that probably is due to the action of interferon gamma produced by the infiltrating cells. So we were expecting a relatively high level of PD-L1 expression in the thyroid follicular cells. And we look for it, and here you have a summary of the results and representative micrographs. In the left-hand side, you have tissue from multinodular goiter, in the center from Graves' disease, on the right from a case of Hashimoto. And indeed, there is some PD-L1 expression on the thyroid follicular cells. If you look at the center of the slide, you see some of the cells have between a purple and violet color, which is what happens when you have a mixture of PD-L1 and TPO. And you see two things in this, which is a representative, is that is not as widespread as you would think, and it does not coincide very much with the expression of HLA class 2. So there is only a moderate expression of PD-L1 in the target tissue. We wanted to see if indeed PD-L1 could be induced in vitro in the thyroid follicular cells. The other ligand of PD-1, which is PD-L2, is also, we also investigated it. And indeed, PD-L1 can be induced, and it is expressed even in the glands when you get them directly from the patient. But the spontaneous expression is relatively, the in vivo expression is relatively low. It is spontaneously expressed when you put it in culture, and if something was repressing it in vivo. And PD-L1 is not so much expressed. So the thyroid follicular cells have the capability of expressing it, but they do not exert this capability in full in the thyroid autoimmune tissue. So now we have another piece for that whole hypothesis quite a few years ago. So if nothing else, I can claim that I keep on working on the same subject for a number of years. So we have hit another milestone in our progress to understand thyroid autoimmunity. The summary of the findings on peripheral tolerance in grave disease is in this slide, and it says like that: normal parenchymatous cells, in our case thyroid follicular cell, express normally low levels of HLA class 1 and no class 2, no adhesion molecules, and the tissue does not contain high endothelial granules. And this is one of the mechanisms of immunological ignorance, and is one of the mechanisms of peripheral tolerance. In thyroid autoimmune disease, this situation is reversed, and that facilitates the contact between the autoimmune tissue and the immune response cells. We have recently demonstrated that T cells infiltrating autoimmune thyroid glands express PD-1, which is not so unexpected. But what happens is that PD-L1 is not as expressed in the thyroid follicular cells as we expected. And we can postulate that the progression from focal thyroiditis to clinical autoimmune disease is normally restrained by this negative checkpoint ligand as PD-L1. And when we administer negative checkpoint blockers, we unrestrain, we enhance the response, we unblock this mechanism, and this is why we get thyroid autoimmunity often as a complication of this type of cancer immunotherapy. And finally, as I said, the insufficient induction of PD-L1 and other negative checkpoint receptors may be a crucial factor in the development of the normal autoimmune thyroid disease. Now I would like to discuss a little bit about central tolerance. To discuss it, we have to remember which are the main antigens that play the central role in the autoimmune response. In the colloid, we have the thyroglobulin, which is a deposit for the thyroid hormones. In the thyroid follicular cells, in the apical pole, we have thyroid peroxidase, which is an enzyme that catalyzes the iodination of the thyroglobulin and the tyrosines. And then we have TSH receptor, which is in the capillary pole of the thyroid follicular cells. This TSH receptor can be, to some extent, shed into the circulation. From what we know in general about how central tolerance works, we will expect that thyroglobulin, TPO, and TSH receptor will be expressed in the medullary thymic epithelial cells, and then will be the T cells capable to react with them will be deleted, and well, not completely obviously, but the frequency of the autoreactive T cell to this antigen will be drastically reduced by this effect. One intriguing finding in which we participated, but we were anticipated by other researchers, was the finding that polymorphisms in the TSH receptor are associated with risk to Graves' disease. And if we rank the genes that predispose to Graves' disease, TSH receptor is among the main ones. And that's relatively not unusual, but it's not that common that a target antigen, the polymorphism in a target antigen, is a predisposing genetic factor in an autoimmunity. So our hypothesis was, this was with Roger Colobran. He is the one who spotted the SNPs in the intron 1 that were associated with risk to Graves' disease. And we postulated, following the ideas that we have developed with Dolores Quemada many years before, is that maybe there was central tolerance to these thyroid antigens, and what may happen is that the alleles that predispose to thyroid immunity, the TSH receptor alleles that predispose, will do that by reducing the amount of expression of that particular allele in the thyroid, so that the alleles will influence the expression of TSH receptor in the thymus. This way, influencing central tolerance to TSH receptor. This was not easy to approach, but Roger had the ingenuity of using allele-specific PCR, and by allele-specific PCR, he was able to show that the alleles that contain the protective the protective alleles are more highly expressed in the thymus than the predisposing alleles. And here you see the summary slide in which you see that the ratio of the protective allele to the predisposing allele in the heterozygous people is above 1, almost close to 2, while other alleles that are not associated have a neutral, they have a neutral 1-to-1 ratio. So what's so special about TSH receptor and why it predisposes to thyroid autoimmunity, and even why so often we get antibodies that are stimulating? Because in autoimmunity we have many autoantibodies, but stimulating, so they are capable to mimicking the effect of a hormone or of an hormone that for millions of years evolved the right fitting of the hormone into the receptor. That we develop an antibody that has the same property. It is puzzling. If we look at the TSH receptor, well, it reminds us a little bit of a toll-like receptor, and it is unusual about it that it has a hinge and is connected, the extracellular part is connected to the transmembrane by a connecting peptide and two disulfide bonds. So it has a little bit of a special structure. It has a constitutive activity, and it seems that activation depends on flexing the receptor when it binds either to the antibody or to the thyroid. It's also a bit amazing that you get thyroid stimulating antibodies, for instance, in people treated with immunotherapy, with alemtuzumab in multiple sclerosis, but you get them in a high frequency, and as I mentioned, in a proportion of cancer patients treated with negative checkpoint blockers. Even in those, you get more often hypothyroidism. So one of the things we did first, it was to check whether this TSH receptor gene was expressed in what it was expected to be expressed, which is in the medullary medullary epithelial cells. And to our surprise, that was not the case. While TG and thyroglobulin and TPO was expressed in the cells of the medulla that are responsible for negative selection, TSH receptor was mostly expressed in double positive thymocytes, so in the maturing thymocytes, in the maturing lymphocytes. That was flabbergasting, unexpected. The first thing we wanted to do is to check whether that expression was functional, and Mireia Jimenez Barcons did a lot of work in our laboratory and demonstrated, and other authors demonstrated as well, that actually these immunoglobulins are capable of interacting with the thymocytes and stimulating them. And actually, there is a clinical correlation to that, because people with Graves' disease, they also have thymic hyperplasia, as you will see in the B panel on the TAC scan on the right side of the slide. And when you treat them, when you treat Graves' disease, the thymus shrinks. And it is known that with treatment, people with Graves' disease, the level of antibodies to the TSH receptor also drop. So what is going on in the thymus of a Graves' patient? We really don't know, and we will make some hypotheses later. But one of the things that Roger was very cross was that the English group that had described the association of the polymorphisms of in the intron 1 with thyroid autoimmunity have postulated that these polymorphisms will influence the proportion of the different isoforms of the TSH receptor. The TSH receptor has several isoforms. I didn't tell you that, but that's not so unusual in many genes, right? And in particular, they postulated that this allele, the predisposing allele, will favor the production of the soluble form of the TSH receptor, and that soluble form, by going to the periphery, will favor the production of antibodies. In the first place, as a general immunologist, we could not really see why a soluble molecule in a non-inflammatory context circulating in the periphery will trigger autoimmunity, because then we will have autoimmunity to every soluble protein, right? So we believe that our hypothesis that was a differential effect on the expression of the gene in the thymus was close to be true. But then he convinced Anna Marin, who was then a young faculty in our laboratory, to dedicate her thesis, her doctoral thesis, into disproving the hypothesis of Brand and Gogh. Here you have the structure of the gene. You need to understand a little bit the structure of the gene to understand the next few slides. So we have, it's a gene which is particular, has this very large intron. It has all these repeats that are here. They are where the hormone binds. You have the hinge, and then you have the transmembrane. And at the end, this is, I didn't mention it, is a GPCR, so it's a gene that then binds a G protein, and this is the way that the signal is transduced. And it's a complex signal, and there is quite a bit of controversy how many pathways are affected by the activation of this receptor. But indeed, we have two isoforms that are shorter, that are called short ST4 and ST5, that are produced as message, and they are produced probably as protein. There is not so much evidence. So we wanted to show, we wanted to see if actually the alleles influenced into the production of the isoforms. And Anna spent quite a little bit of work and hours trying to see what happened. And I will go through it because this is very technical, but basically she showed that actually the alleles in the intron 1, the alleles that predispose to thyroid immunity, do not influence on the production of the TSH receptor isoform, neither in the thyroid, neither in the thymus. And we did that experiments with an overwhelming higher number of glands that the British group did. But then, when we look closely to the results, and actually it was Anna who spotted it, they say, well, there is a lot of these isoforms in the thymus. We expected that there will be very little, but actually there is quite a bit of TSH receptor in the thymus because it's expressed in so many thymocytes. This relatively high level of expression actually explains one Western blot that was carried out in our laboratory many years ago by Maria Jimenez Barcons, in which she detected quite a few TSH receptor, holoreceptor, and other subunits in the extract from the thymus. And at that moment, we were surprised that was so much. But then now we know why it is, and now we can also see even that in the thymus there is expression of thyroid receptor, and we can see also that some of these thyroid receptors sometimes can be seen within macrophages and within dendritic cells. And all these findings, especially this relatively high expression of the isoforms of that are soluble in the thymus, led us to consider the possibility that there is TSH receptor split tolerance, or split tolerance to the TSH receptor. This is a concept that is not new in autoimmunity. We know that in the case, for instance, of myelin basic protein, there is a split tolerance. There is tolerance to most of it, but we lack tolerance to some particular epitopes that are not expressed in the thymus because differential isoform expression in the thymus and in the periphery. This differential expression of isoform affects also some of the type 1 diabetes autoantigens, and we know that T cell in the periphery, in both cases, is directed against those epitopes that are not properly tolerized in the thymus. So we try to think if that could be applicable into our situation. We have no proof, but we can imagine that double positive TSH receptor thymocytes will die in high numbers in the cortex because all the double positive thymocytes do. They would be then taken up by thymic macrophages, which are cells that not take, in principle, part in negative selection. And therefore, full-length TSH receptor will not be reaching the areas in which negative selection takes place, because we know from the experiments on that selective investigation of the transcription of these antigens in MTEC that I already presented previously, the TSH receptor is not expressed in a detectable way by medullary thymic epithelial cells that are the main negative selectors. But we can think that the soluble part that will be produced by the very abundant double positive TSH receptor positive thymocytes in the cortex will be taken up by dendritic cells, and that will be diffused even into the medulla. And these dendritic cells do play a role in negative selection. So we will have a situation in which we will have at the T cell level tolerance to part of the extramembrane, part of the receptor, but not full tolerance to the full receptor. And what happens in the periphery? Well, we know from general knowledge that B cells are not so extensively negatively selected in the bone marrow, so in the periphery there must be quite a few B cells capable of recognizing any parts of the TSH receptor. And if those B cells take up TSH receptor, either the soluble part or the whole part, because there is some necrosis or there is some destruction of the thyrocytes, they could get help from the T cells that are specific for the transmembrane part, for the parts of the TSH receptor that have not been tolerized in the thymus. This help from the T cells can result in the production of antibodies, and the antibodies will be mostly directed to the extracellular part, which is the one that are more accessible to the B cells, and those will be the stimulating antibodies. This is just to show, it's a better picture than the previous one actually, in which you see that there is some expression of the TSH receptor in the cortex of the thymus, and that is expressed. We have demonstrated that it's in the double positive cells. So at this point, last year, we have reached another milestone in our research, and we have a new hypothesis on how the TSH receptor stimulating antibodies may be generated. And we have also an hypothesis on why autoimmunity to the thyroid in general does not happen all the time, because it's in part prevented by PD-L1, but also why happens relatively often is because PD-L1 is not really so actively expressed in the thyroid immune glands. So we were in an interesting point, and this is a little bit of the summary. We have now these two hypotheses to work on, and we carried on. And initially, one of the things we wanted to prove was that actually PD-1, PD-L1 axis could play a role in presentation in thyroid follicular cells to T cells in their interaction, and we set up experiments co-culturing intrathyroidal lymphocytes with the thyroid follicular cells from Graves' glands patients and trying to block it. And then we also did experiments as a control with stimulating the T cells and seeing the effect of the thyrocytes on them. And we were doing that with Daniel Álvarez de la Sierra when something that you are all familiar happened, and that was in March last year. And we were confined, but we felt that we had to do something, and actually Manuel Hernández, the present head of the division of Immunology, Vall d'Hebron, started to set up this big biobank of the samples that were sent to our laboratory for IL-6 measurement, and in general for all the measurement of the biochemistry. And I helped him by generating a database that will put together and could give a perspective of what happened to these patients. And now we have all this data, and we got a project from the Carlos III, a project that is in collaboration with Hospital de Bellvitge, with the Division of Immunology, Dr. Jordi Bas, and with the Division of Immunology at Germans Trias, my former hospital, with Eva Martinez Cáceres and Ana Teniente. This is the first analysis just of the Vall d'Hebron data. I am waiting for the data from the other two hospitals that are coming. But this is already a large number of samples, about 1,800, and you can see that is those patients that have a poor outcome. Either they have the severe disease or they die. They have higher IL-6, and most of the acute phase reactants are also fine. It is difficult from this data deduct what is first what sets in motion the inflammatory process. We obviously can do multiple correlations, and we can do principal component analysis and machine learning, and we can expand the analysis of cytokine. But we have this strong suspicion that everything starts there. So you and everybody is wondering what really happens at the beginning, but knowing that the virus is not so variable, and he didn't have that much time to readapt and to change. Given the clear hyperinflammation that we see in critical patients and the high levels of IL-6 and other cytokines from the onset, given the relatively mild clinical features of COVID-19 in most patients with immunodeficiencies or in patients that are immunosuppressed, the recently demonstration that type 1 interferon are involved, at least in a proportion of patients, some because of the genetic defect, some because of the surprising evidence of antibodies to interferon alpha in almost 10% of the patients. By the way, I am very proud that the Vall team played with Dr. Pere Soler and all the team play a role in helping to produce the results. And also the role of age, and aging is clearly the main determinant or the main factor. And we know that there is a decline on the immune system, especially after 70. Especially there is some regulation of the natural immunity and difficulty of mounting an effective adaptive immune system after 70. And also that the most typical comorbidities are type 2 diabetes, obesity, that we know now that they coexist with a relatively low level of inflammation that may disturb the organization of a well-regulated natural immunity. All that seems to point that the immune mechanisms are the main determinants of the severity of COVID-19. Obviously, the size of the viral inoculum probably will also play an important role. But we think it's still necessary, in spite of the many papers. Many of them are in relatively small numbers. Some are high quality, but still, I think we need much more good quality immunological research in SARS-CoV-2, both to treat better the patients and to be able to assess the vaccines and to help to assess who should be vaccinated and which is the best vaccine and which is the best regime for the vaccines that we hope they come soon. And the last and the more sentimentally one slide, I didn't put the people who died that helped me in the first period, like Gianfranco Bottazzo, Deborah Doniak, Emilio Curie, which I really miss. But I will concentrate it in the last few years. In the last few years, there were 3 people that have been really extremely helpful and fantastic colleagues, which are Roger Colobran, Ana María Sánchez, and Daniel Álvarez de la Sierra. The 3 of us have been with me. We have produced these results of the last 10 years. Obviously, during the previous 23 years, I worked very happily with Eva, with Manel, and with Dolores Jara-Quemada and all the team at Germans Trias y Pujol. And I would like to thank also the support of the whole group of the Division of Immunology and of the Diagnostic Immunology group, now headed by Dr. Hernandez. And over the years, I have to say, thank also a number of very active and brilliant master students, Carmen, Paloma, Maria, Nerea, and the clinical collaborators, the endocrinologists like Ana Casteras, Gabriel Obiols, before Germans Trias, Ana Lucas, of course, and the surgeons who help us, Oscar González, Pablo Moreno, and the histopathologist Carmen Iglesias. And that's all. Thank you very much. I'm sure I've forgotten somebody, but I apologize for that. And now I will take questions.